Good evening, everyone. Welcome to the eighth week of live problem solving session for medicinal chemistry. I'm your uh, TA for this course, Gitanjali Singh, a PMRA fellow from Department of Chemistry at IIT Kanpur. This problem solving session is held every Monday from 6 p.m. to 8 p.m. This is the same link which has been valid for a long time now and will be valid for the next uh, upcoming 4 sessions. In this session today, we'll discuss last week's summary, last week's assignment, some modular question and topic for the upcoming assignment. In case of any network disruption, please do not leave. Try to remain online. If it's from my end, I'll try to join as soon as possible. I would appreciate if you guys post your questions on chat box, but if that's if you don't feel like writing and typing the paper, the question on chat box, you can unmute yourself and ask the question then and there. I'll try to explain it. If for some reason I cannot explain the question, I'll note it down and I'll discuss the question in the next session. This presentation and class is recorded, and the link of the session will be shared to you soon. So, on week 8, Sir discussed optimizing drug targets interaction in 4 parts. Along with that, there was a tutorial, which was tutorial number 12. So, let's move ahead and discuss last week's assignment. The the answers to this assignment has already been published. I'm just discussing this just to give you an idea of how to solve the assignment, and if there is anything you don't understand, you can ask me. So the first question was, what is the term used for drugs such as sildenafil, and this is the following 4 drugs, which are all developed as antihypertensives following the discovery of a very popular drug, captopril. Now, the structures of the drugs are given, and you were given 4 options: me too drugs, orphan drugs, analogs, and fourth, active principle. Yes. Yeah, sure. Hanji, I'm sorry. Oh, my screen is not visible. Oh, I'm sorry. Wait. Just a minute. Is it visible now? Okay. The thing is, I'm recording just a part of the question. So, is that a problem? Is that somehow creating problem with the resolution or anything? Please let me know. Okay. Perfect. So let's discuss what these terms are, the terms that are given in the uh, options. Me Too drugs. These are chemically similar to an already existing drug that may have, you know, this Me Too drugs are like, uh, uh, so suppose there is a drug already in market. So Me Too drugs are chemically similar to that drugs with a little bit modification. Now this modification is done either to increase the efficiency of the drugs or to lower the side effects and increase the pharmacokinetic effect. Generally to alter this, but it's more or less the same. So there are three criteria like the chemically similar to already existing drug with very minor modification. It's developed to compete with established drug in the market, obviously, because obviously it's very similar. It often has similar efficacy and mechanism of action, but it differs slightly in pharmacokinetics and side effects. So even slightly lower side effects could make the drug a new drug. For example, ibuprofen or naproxen. Now, these two have similar action, but they have slight difference among themselves. Orphan drugs are the drugs that are developed for rare diseases and that affect a very small population. You know, they often lack commercial viability because it's a rare this medicines are for rare diseases. Only a small population of humans are affected by this, so they are not really marketable, and companies don't really get benefits from them, so they often lack commercial viability. The government generally provides incentives to encourage R&D for these drugs, but obviously because they don't have any commercial profit, so it's not a very lucrative investment for the company to just do it for the sake of, you know, goodness of their heart or anything. So they are often called drugs. The next term was analog. So these drugs are like structurally similar to other drugs and molecules, but have difference in functions and potency. They're often designed to enhance efficacy, reduce side effects, and improve bioavailability. But you have to understand, Me Too drug and analogs are different. Me Too drugs are the drugs that are slightly modified version of an already existing drug in the market. Analogs have similarity in structure, but their functions can be different. Their potency can be different, and so that's the key difference here. They're often designed to enhance efficacy, reduce side effects, and improve bioavailability. So, insulin analogs like insulin Lispro and regular insulin, these are examples of analogs. Active principle, the main active principle, so it's like what is the component of a natural you know how to say, so if there is a drug, a naturally occurring thing, so what is the main active component of that natural product? That's called active principle. The main bioactive component in a drug or a natural product that is responsible for the pharmacological effect, that's called active principle. It's found both in synthetic and plant-based medicines, and examples can be morphine or caffeine. So is that okay? uh, the slides are not. Are my slides visible? Okay, I'm sorry. Is it visible now? You're welcome. So, should I repeat everything again or should I move ahead? So, okay. So, now that we know what the four options are, we have to understand the question. What is the term used for a drug, the following drugs, all of which were developed as antihypertensive following the discovery of captopril. Now we know what all these drugs means. So what what is the main answer? The accepted answer was Me Too drugs. These drugs, the term used for such drugs is ACE inhibitor. Why? Because these drugs work by inhibiting angiotensin converting enzyme, that is called ACE enzyme, which is responsible for the conversion of angiotensin 1 to 2, a vasoconstrictor. Now, vasoconstrictors are the one that constrict the our vessels that carry the blood pressure. So, by constricting it, you can increase the blood pressure. Think of it as a pipe. So, a normal pipe in which water is flowing. When you constrict it, the pressure of the fluid in the pipe increases. So that's called constriction. That would increase the pressure in the inside the vessel. And then vasodilation is when the pipe dilates. So the amount of fluid passing through it, we assume the volume is constant. So this constriction and dilation affects the pressure of the blood in the pipe. So these drugs work by inhibiting angiotensin converting enzyme, which is responsible for conversion of angiotensin 1 to 2, which is a very potent vasoconstrictor. By blocking ACE enzyme, this drug reduce blood pressure and are commonly used as antihypertensives. They are developed. They were developed following the discovery of. Oh, so is there any? So my slides are still visible, right? Okay, thank you. So why do we use the you might have noticed that all this all the name of these drugs have like pril in the suffix. That is common. So this is just to identify and classify this drug within a group for easier identification. So the suffix pril is a common naming convention used for ACE inhibitors to indicate their pharmacological class. This helps in easy identification and classification of drug within a group. So hypertensive drugs are antihypertensive drugs, obviously, are you know eh, classified in many other classes. The one that we discussed was ACE inhibitors. These medications help relax blood vessels by preventing the production of angiotensin 2. So if angiotensin 1 is not converted into 2, they so this so this is a vasoconstrictor, remember. So if it cannot constrict the vessels, the blood pressure won't increase. And if the blood pressure is not increasing, so that's how it's controlled. So yeah, that's first class. The second class is angiotensin 2 receptor blocker. Now they also work by relaxing blood vessels, but instead of blocking the production of angiotensin 2, they block the effects on the blood vessel. The third class is calcium channel blocker. These medications prevent calcium from entering heart muscles and blood vessels, which helps relax the blood vessels and lowers the blood pressure. The example you can see here. Uh, the common, a very famous example is, I think I might have confused. I confused it with something else. It's okay. The fourth is diuretics. These are called water pills. Now, diuretics help the body rid itself of excess water and salt, which lowers the blood volume and blood pressure. And the fifth one is beta blocker. Beta blockers slow down the heart rate and reduce the force of heart contraction, which in turn reduces the blood pressure. So these are the five. Okay, there are three more classes. Alpha blockers. Alpha blockers also relax the blood vessels, making it easier for blood to flow through them. The seventh one is centrally acting antihypertensive drugs. These drugs act on CNS and reduce sympathetic tone, which leads to decrease in blood pressure. And the final one is vasodilator. Now, vasodilators relax the smooth muscles of blood vessel, causing them to widen and lower the blood pressure. So, these are the main classes of antihypertensive drugs, but these drugs are not exclusive. It does not mean that because they are vasodilators they are only antihypertensive drugs. Drugs have various classes, and several drugs can fall into different classes, and these classes are not very exclusive class. One drug can be a vasodilator and at the same time act as supplement for something else. So these are very general and very inclusive class of classification. Now let's move to the next question. This question was match the following question, where we had to match the group 1 with group 2, in which they had given the routes of administration and their preferred sites. So group 1 had subcutaneous, intrathecal, intramuscular, and intravenous. And number 4 was different sites: spinal cord, surface of skin, muscle, and vein. We had to you had to match them from their preferred site. So let's first discuss what subcutaneous and what all this routes of administrations are. We have discussed this in my previous class as well, but we'll discuss this thoroughly here. So subcutaneous injection. So subcutaneous injection delivers, so it's basically a layer between the skin and just under the skin, and before it reaches the muscle, so in the thermus, so a fatty layer just below the skin. That's where these injections are injected. It delivers medicine into the fatty tissue layer under the skin, allowing for slow and steady absorption of medication. You can see the sites shown here. And these are where we have small fatty tissues just below our skin. This method is favored for medications that need to be absorbed slowly into bloodstream, like insulin, blood thinners, and certain hormones. These are the common sites where these subcutaneous injections are generally given: anterior and posterior. The second option was intrathecal. Now, intrathecal injections are given into CSF, cerebral spinal fluid. So this involves giving the injection directly into CSF surrounding the spinal cord or subarachnoid space, bypassing the blood-brain barrier. We've talked about blood-brain barrier in our last class thoroughly, so I'm not going to repeat it again here. So this bypasses the blood-brain barrier for targeted drug delivery, often used for pain management, anesthesia, and certain type of chemotherapy. You can see the location of intrathecal injection to thecal sac. This is where it's generally given, and we know our spinal cord is floating in the CSF, and that's how it reaches it crosses the blood. So in order for medicine to cross blood brain barrier, Generally, medicines injections are given in this intrathecal route of administration. Now, intramuscular, it's directly injected into muscle. It's generally preferred because it's preferred over subcutaneous because it's faster. Subcutaneous is for slower absorption and sustained absorption. Muscles, when you inject anything into muscle, it has comparatively faster absorption as compared to subcutaneous. And yeah, it's not subject to first pass metabolism as in oral administration. So common sites are deltoid muscle and gluten muscles, and there are several other examples as well. And the final one was intravenous, when the injection is directly injected into veins, so that it has, if you remember, this has 100% absorption, and this is how we calculate bioavailability as well. So yeah, if you want 100% absorption and very fast, it's generally given intravenously. So the correct option was subcutaneous, surface of the skin, intrathecal spinal cord, intramuscular muscle, and intravenous vein. So you can see 1 to 2, intrathecal 1, B to 1, C to 3, and D to 4. Now let's move on to the third question. Which of the following is correct definition of bioavailability? 1. Bioavailability describes the portion of a drug administered that is metabolized very quickly and thus is not available to induce a physiological effect. Number 2. Bioavailability describes the ability of the administered drug metabolites to cause undesirable physiological effects. Third, bioavailability is used to describe the fraction of a dose of drug administered that is present within the body and facilitates the desired physiological effect. And the fourth one is bioavailability is the length of a time an administered drug is present in the body and is thus available to cause a physiological effect. So by the definition of bioavailability, we know that it's the amount of drug um, that's available in the body, to show the physiological effect. So again, I'll give the example. So if 100 mg is a dose of the drug that is given to a patient, after administration, if the body gets 100% of the drug, if the body has like 100 mg still in its system, it's called bioavailable drug that is responsible for causing the physiological effect, that kind of effect that we want. But in case of oral administration, that does not happen. The gets metabolized. The drug gets metabolized, and so the bioavailability becomes less and less. So yeah, going by definition, the correct answer is the third one. Bioavailability is used to describe the fraction of a dose of a drug administered that is present within the body and facilitates the desired physiological effect. Let's move on to the fourth question. Which of the following is not classed as an in vivo test? 1. Testing anti-cancer drugs on a tumor grown on the back of a rat. Number 2. Testing analgesics by determining a guinea pig's sensitivity to pain. 3. Testing antibacterials by measuring the growth of microorganism on petri dish in their presence. And number 4, testing antibacterial agents in infected rats. Now, in vivo, in vivo means in the body. So there are three kinds of experiment broadly: in silico, in vitro, and in vivo. In silico is done in on computers and simulation and things like that. So it's performed in a virtual setting on a computer or a virtual set. In vitro is in glass, meaning the study takes place in a test tube or a petri dish, something like that. In vivo, that means in life. That means study that takes place inside an organism. I mean, in an organism. So that's that's broadly uh, what the difference is. So among the following option, the third one is talking about in vitro rather than in vivo, testing antibacterials by measuring the growth of microorganisms on a petri dish in their presence. That's in vitro, not in vivo. So that's why it is not classed as in vivo. So the correct answer was third. So let me discuss this in more details. What is in vivo and in vitro? So what exactly are in silico experiments? Like in general sense, how can we perform biological experiments on computer? Well, there are several um, kinds of things that can be done by virtual simulation, and just by having a largest data set of obviously datas that we have, and by focusing our attention on what we want, what kind of information we want from the study that we are doing. So in silico studies of biological experiments are carried out entirely on a computer or via computer simulation. This is the newest of the three branch. The first one was done in 1989. So these studies have already contributed quite a lot in biomedical research and everything. For example, in there are many studies. So in so there are three more. We'll talk about three varieties. I've collected three techniques. The one is bacterial sequencing technique. These methods are done having deployed to identify bacteria through bacterial DNA and RNA sequencing. These are mostly used in PCR, polymerase chain reaction. The second one is molecular modeling. So this is the science of representing molecular structures numerically and simulating their behavior with equation and quantum and classical physics. And the third one is whole cell simulation. So this refers to computer models of cellular behavior. So what they do is they basically try to mimic and try to uh, simulate the conditions in which the whole cell is considered as an entity, and how small modifications can lead to what kind of changes. Those are the kind of studies um, that is observed, and they take data out of it, and yeah, that's how they simulate things. So more recently, artificial intelligence technology are gaining prominence along with whole target discovery process. Deep learning, machine learning tools allow researchers to automate and extract more meaningful information from experimental outputs in order to generate models and build complex networks by integrating data sources. So because we have like many databases now dedicated to particular and very organized studies, having the data of protein structure and bacterial sequence and every kind of sequence. So if we are sure and if we know how to actually read this data and utilize this data for a targeted study, we can basically build a model and try to simulate a situation in which we can test how one single change can affect the entire, you know, um, the entire cell per se, or the bacteria or sequence, and how the change and what kind of mutation could lead what kind of response in the cell. So that's basically what is what is done in silico. Now, in vitro assays, so these are the one that we know about, like um, minimum inhibitory test, time kill kinetics, and things like that. So these are done within a glass, this and in a very controlled environment. So they refer to work done with microorganisms as well as cells, tissues, and other biological components that have been removed from a living organism of interest, and basically cultured in petri dish or test tube. These approaches are suitable for cellular and molecular studies, such as understanding signal transduction and molecular signaling pathways. So this provides very cost effective, time efficient, and they do not need any animal models. So they allow for rapid development of new treatments, since many drugs can be studied at one time. That's why they are often the first test of, like, in experimental condition. What happens is, once a drug is designed, once you have a library of molecules, the first thing they do is in vitro test. They check for activity. That's why it's very beneficial. So making a design computationally and getting a hit is still easier, because you have large data set. I won't say it's easy, but that's still the first step only. If our after getting the lead and designing the molecule and making the perfect molecule, if it still has toxicity, and if it does not have any activity, that's where the entire experiment stop. The drug does not come into a reality, does not translate into a reality. So we have to have some activity, and that activity is checked in vitro. The first one is minimum inhibitory concentration, and for them to check MTT assay. So these are the kinds of things that is done in vitro, certain assays. And then after like a good having good results from in vitro experiments, then we go to in vivo, and that's called animal model and things like that. And that's where we start experimenting inside living organism, mainly in mice, and then in pigs and monkeys. And furthermore, you can see the comparison here. In silico is low cost, in vitro is also low cost, in vivo is quite expensive and has ethical issues to it. We don't have to face any ethical issues in vitro conditions and in silico, but again, they don't give us the result. We don't know what a drug can do. There's so much. So that, because it's all in controlled environment, and you don't have other parameters affecting it, so you don't know. Even if a drug has a very good activity in vitro, it might have like many side effects, not side effects, but it might interfere with several other pathway and metabolic activities in vivo. So that's why in vivo is very important. It helps us better evaluate the safety, toxicity, and efficacy of a drug candidate in a complex mode. So yeah, that's why all three are important. The first one in silico is new, but it's very good. It just helps narrow down our hit molecule. So it's very important for drug optimization. Now let's move to the next question. Which of the following statement is true? 1. Parallel combinatorial synthesis involves synthesis of a large number of compounds using the same reaction sequence, where there is a mixture of compounds in each reaction vessel. Number two, parallel combinatorial synthesis involves the synthesis of large number of compounds using different reaction sequence where there is different single compound formed in each reaction vessel. 3. Mixed combinatorial synthesis involves the synthesis of a large number of compounds using a variety of different synthetic routes to produce a mixture of compounds in each reaction vessel. Number 4. A parallel combinatorial synthesis carried out in a specified number of vessels will produce less compounds than a mixed combinatorial synthesis. So let's see what combinatorial synthesis, parallel combinatorial synthesis, and mixed combinatorial synthesis uh, are. So in mixed combinatorial synthesis, there is mix and split. I'll explain it further. The correct answer is, so it was asked which of the following statement is true. So, a parallel combinatorial synthesis carried out in a specified number of vessels will produce less compounds than a mixed combinatorial synthesis. That's true. Now, let's see why is that so. Is there any question till now? Yes, sir. Okay. Okay. So, in combinatorial synthesis, what happens is uh, there are many, so you have, like, suppose say A, B, C, D, you have four things, they are like combined to a raise and something. What we do is you mix everything with everything, and so there is no particular sequence. There is a vessel, you put this and this, so there is no structured way in which you get a combination, and after that you split it and you add this with the next one, and there is no particular sequence, you just mix and split. So combinatorial synthesis is a chemical synthesis method that helps prepare large number of compounds in a single process. That's true. Split synthesis method, again, because you are after one thing you just take it out and mix it with the another set of like A, B, C, D, so you mixed each with everything. Now you have a big vessel A, B, C, D. Now you take up a fraction and you separate them, and then again mix with A, B, C, D. So there is large number of things happening. It's very messy, and you have so many things mixing up, and you just split it. You don't know which order they are forming, whatever. Many types present in a single reaction vessel, and requires a deconvolation procedure to identify individual compounds of interest. So the main target of this synthesis is just to find whether the target is just to see if it has any activity. Once you get activity, then you try to figure out, okay, what is the, you know, how you identify the individual compound. In parallel synthesis, it's much more organized. So in parallel synthesis, what happens is you have A, B, C, D, and suppose you have another thing, so you mix A, B, C, and D with one B, and then you put another reactant, suppose E, F, G, H. You then know the sequence, it's well organized, and you know what kind of product is forming. So it's one bead one compound method and performed in individual compound. Only one type of support bound compound in a reaction vessels, so you know what kind of compound it's forming. It's either support based, solid phase organic synthesis, or solution based. You can just wash away the unreacted compounds. So the common thing here, you'll see large number of similar compounds, obviously, because you're mixing them. Again, automatic, it can be small scale. raisin if it's support, because it's supported, so it's a raisin beads. It's done in on raisin beads. Same reaction sequence applied to many compounds. That's right. But because we basically mix and split in combinatorial synthesis, it's in the same reaction, you're mixing, you're splitting, and then you're putting again and you're mixing. So that's why, combinatorial synthesis, it's very messy. And you have larger library of compounds. In parallel synthesis, it's much more organized, it's less messy, but again, it has smaller library of compounds. And because it's like 1 1 1 1, so different reaction vessels have different compounds, so it's much more organized. So now you understand why this answer is correct. Now let's move on to the next question. Which of the following statement is false with respect to NMR screening to detect drug target interaction? 1. The procedure relies on small molecules, drugs. So when they're talking about small molecules, it's generally drugs, and when they're talking about large molecules, they generally mean the targets. That means proteins. So the procedure relies on small molecules having shorter relaxation time than large molecules. That's the target. Number 2, the procedure can be used on mixture of compounds. That's okay. 3, the method can detect weak binding. 4, the method can identify small molecule binding to different regions of the same binding site. So the correct answer, obviously, so they are asking about what is false. All these three are right. The first one is wrong. That's why this is the correct answer. We'll see why. The false statement is the procedure relies on small molecules having shorter relaxation time than large molecules. That's wrong, because generally small molecules have longer relaxation time compared to larger molecules. When a small molecule binds to a larger target, its relaxation time decreases, due to which due to the slower molecular tumbling and increased interaction. So the principle of used with this technique is SDD NMR, saturation transfer difference, and ligand observed NMR. These are used to study drug target interaction. So why are the other three true? The first one, the procedure can be used on mixture of compounds. That's true, because you know they actually do this. Sorry. They actually do this for screening purpose. That's sorry. Yeah, so that they are used for screening purpose, so they can be used in a mixture of compounds. We'll discuss how they do it. The method can detect weak binding. That's true. NMR is highly sensitive to weak interaction, and it detects even on like millimolar, micromolar range. So that's true. The method can identify small molecule binding to different regions of the same binding site. That's true, because you can see that from chemical shift and NOE. So that's one entire new concept on its own, but it can basically sense the neighboring difference or change in its neighboring interaction. That's called nuclear overhauser effect. It's a very deep and a very big topic on its own, so has explained it already. So yeah, now let's see what this how they are using NMR for drug interaction screening. So uh, think of a drug like the small molecule and as key, and the target molecule as a lock. So in order to find the cure, to find a proper drug, you need to have right key for the for the lock. So right key for the lock. So how does that help? So basically NMR helps us to listen what the how the molecules is interacting in the solution. Now the small drugs, they can move freely in the solution. They can spin, they can tumble, spin, vibrate, and they can move around easily in the solution. And when they do that, they give very clear and sharp signals. Wait. Now, once the scientists, if they add the lock, if the key is right, it'll go and get to the lock, right? And it can basically create the difference there. So that would lead to change in the chemical shift and the chemical behavior, the spectrum that we see, that would change. If a drug binds to the target, it slows down the free moving drug. It will slow down. If there is binding, that would change in the resonance pattern or in the NMR, and that would be reflected in the spectrum. If there is no binding, the small molecule remains unchanged. We talk about small molecule because it's easier to see their spectrum in the NMR spectrum of protein. So if you're talking about fluorine 1H NMR, it's very difficult. That's not used. Basically, they use fluorine or phosphorus NMR for this. Sometimes they use 1H as well, but it's very very complicated, and it's only used for very specified thing, like the kind of spectrum that we know about. Because you can imagine proteins have very complicated spectrums, NMR spectrum. So yeah, that's why. And smaller molecule will have like easier to read spectrum. I'm not saying that's the only reason why they use it, but that's one of the major reason why they used it. So if the drug binds well, there's change in NMR shift. The NMR shifts and disappears and broadens. If the drug does not bind, the signal stays the same. This helps us identify promising drug candidates without needing to label or modify the molecule. That's why they're used for screening. NMR is useful because this can test many drug at once. Obviously, that's why the other option was right, screening of mixtures. It can detect very weak interaction. Again, that's very good because that's what we are doing, right? And can map exactly where the drug binds in the protein. Let's move on to the next question. What is a term used for an animal that has been genetically modified for in vivo test? Again, we discussed what in vivo and in vitro are. Yes. So, is there any question? Yeah, yeah, that's correct. That's a correct answer. The correct answer is transgenic. So, in vivo means in body. So they are asking the term that is used for an animal that has been genetically modified for in vivo test. So let's see what all the other four options are. What do we mean by chimeric animal, transformed animal, hybrid animal, and transgenic animal? So um, chimeric animal. So chimera is an animal that contains cell of two or more genetically distinct sources. So sources, that means where they come from. Different cell population originate from two different zygotes. I'm saying two, it can be more than that, like fusion of embryos or transplantation of cells. So a mouse generated by injecting embryonic stem cells from one strain of blastocyte or of another. So that's basically chimeric. It has a different genetic composition due to its genetic source. Now, transgenic animal has a foreign gene. So it's so suppose um, uh, you have a packet of uh, so you have a vitamin D3 bottle, and in those bottles you have vitamin D3 capsules, but suddenly one of the capsule is replaced with a magnesium supplement. They have a foreign gene introduced to a genome via genetic engineering. So that foreign gene is in fact quite stable and has integrated and become a part of their DNA, and can be inherited by the offspring. That's how they do it. So a mouse expressing green fluorescent protein GFP due to insertion of a jellyfish gene. So they just introduce a foreign gene in a genome of an organism that can be transferred from one generation to the other one. They are called transgenic animal, and they are modified for in vivo experiments. Hybrid animals, obviously, two different species, when they have mated or subspecies, they create a different animal. They are called hybrid animal. They don't belong to one specific subspecies or species. So it's a result of either natural crossbreeding or artificial crossbreeding of two genetically distinct individual, like a mule, that's a cross between donkey and a horse, and a liger, that's a cross between lion and a tiger. And transformed animal. So yeah, so it's not for animal exactly, it's more for bacteria. So a transformed animal would typically refer to an animal which will have cell that has gone genetic modification, often through viral and plasmid mediated gene transfer, and that does not generally happen in animal. They are generally done in bacteria, while uh, genetic yeah, so viral transfer. So they have plasmid transfer, they cut the plasmid and they transfer one gene from one bacteria to the other. So you get the sense, right? That's a transformed animal person, because they're talking about animal, so it's animal. For example, cells of a transgenic mouse transformed using a viral vector to express a certain gene. So this is simple different the key differences in simple terms when they're talking about chimeric mix of cells from different genetic source in the same organism. Transgenic, it's a genetically engineered animal with a foreign gene that has been stably integrated in its genome. Hybrid is a crossbreed between two different species or subspecies. And fourth, transformed, an animal or a cell that have undergone genetic modification, that's usually temporary. So that's called transformed. So the correct answer is transgenic. So the term, so we know these are transgenic animal, we use them for in vivo studies, but why? So genetic modification, they have like foreign genes that are called transgenes. They are generally inserted to the genome of an organism via techniques like microinjection, viral vectors, via they use phages to transfer genes from one organism to the other one, bacteriophages, and again CRISPR Cas9. This is a very interesting technique. uh, you might have read about it, and if you're interested in knowing more about it, you can tell me, we can discuss this in the next session. Again, this integration has to be very stable. You cannot just integrate some foreign gene and expect it to be part of the genome. Okay, so we can discuss this in the next session. I can prepare a few slides on this technique, and we can discuss it before starting the session, if that's okay. And the integration of the foreign gene has to be stable integration. It has to express itself and transfer from one generation to the next. That's a very important criteria, and it's used for gene study, study of the gene function, disease model, and drug testing in live organism. So examples are knockout mice where a specific gene is deleted to study like cancer or other neurodegenerative disorders. Humanized mice, a mice engineered to express human genes for biomedical research. And glowfish, this is what we talked about, a fluorescent fish modified with jellyfish genes to study environmental toxicity. The next question is, what is meant by lead compound in medicinal chemistry? So we went through this question, like different form of this question since her first assignment. A drug containing the element lead. No, that's not what it means. A leading drug in a particular area of medicine. 3. A drug that acts as the starting point of drug design and development. And fourth, a drug which is normally the first to be prescribed by for a particular element. No, the correct answer is 3. Yes, sir, sure, you can leave. It's okay. So the correct answer is a compound that acts as a starting point for drug design and development. Lead compound is the one that shows some kind of activity around which we plan the it's basically the active scaffold that gives the drug its activity. So if say we are talking about an antibiotic molecule and antibacterial, so the scaffold that is responsible for the action of the antibiotic molecule is what we call lead. We design drug around it to increase its efficacy, and that's like to decrease the toxicity, and we work around that particular compound, that particular scaffold, and that is called lead compound. So a compound that acts as a starting point for drug design and development, that is what is a lead compound. Yeah, the next question is, the drugs that are taken orally pass directly to liver once they enter the blood supply. This is known as the first pass effect. True or false? So, or once we take drugs or any drug orally, it goes through our GI tract, gets to an intestinal tract, it goes to our mouth, esophagus, stomach, and liver. And liver being the main source, liver is the main source of metabolism. Yes. So liver is the main source of is the main place of metabolism. This is where most of the metabolism takes place. Metabolism takes place in the stomach as well. It starts, so the drug starts to get metabolized from our mouth itself, but because liver is the main site of metabolism, so you know um, that's why people often consider liver to be the most important, because that's where the main enzymes are and that's where the major metabolism takes place. So the answer is true. Let's move on to the next question. Which of the following routes of administration is the fastest way to deliver a drug into the bloodstream? 1. Intramuscular, 2. Intravenous, 3. Intrathecal, and 4. Oral route. The correct answer is intravenous route, because it's directly injected into the um, our systemic circulatory systemic circulation. So it's, as soon as it's administered, it bypasses absorption and it directly goes to the distribution. So it does not waste its time getting absorbed and metabolized. So that's the fastest route of administration. Intramuscular route is also fast compared to subcutaneous. So if you want a slower absorption, we generally go for subcutaneous, as in case of insulin. Intramuscular, if you want faster action than subcutaneous, we generally take the intramuscular route. Intrathecal, when you have to directly inject particular injection for brain or to CSF, spinal fluid, and for the drug to bypass the blood to for the blood to pass the blood brain barrier. I'm sorry. For the drug to pass the blood brain barrier, intrathecal route is taken. Oral, the best way, but it it has to go through first pass effect, so it's least bioavailable. That's why, but still the safest and the most preferred way of drug administration. So the correct answer is intravenous route. Now, question 11. Primary and secondary amines act as both hydrogen bond donor and acceptor. Sorry. Whereas tertiary amines are only hydrogen bond acceptor. So you can see this is primary amine. Primary amine, this can be any group R or anything. This does not have to be amide, because we are talking about amines. That's why this group was just taken, because this is the most common way of common, this is basically part of protein amide and things like that. That's why. So CONH bond, when we are talking about primary, N has 2H. When we talk about secondary, that means nitrogen will have at least one H and 2R group. And in case of tertiary, nitrogen will have no H. So if it does not have any hydrogen, how will it be a donor of hydrogen bond donor? It will always want to have like more H instead of like, because it does not have anything. How can it give it? So that's why it does not act as sorry, what was the question? Does not act as hydrogen bond donor. Instead, it only can act as hydrogen bond acceptor. Isosteres of oxygen are S, NH, and CH2. So that isosteres refer to molecules or ions that have same number of valence electrons and similar shape and electronic properties, often used in context of bioactivity and drug development. That's true, but the easiest way to do that is count the number of electrons. If they are same, they have same number of electrons. Perfect. So this is true. We discussed about Me Too drugs, thoroughly in, during the start of our lecture. So the term Me Too drugs are used for drugs that are similar in structure to a known drug, which is used for different purpose. So if you remember, in our starting class, we discussed what Me Too drugs are. I'll just go to that slide. Me Too drugs, they are structurally, so suppose they are just a simple modification of an already existing drug in the market. They have same function. They don't have different function. They have literally the same function. It's just that they have different, they might have better efficacy, they might have like better activity, or lower side effects. They're just, that's why they're me too. They have often similar efficacy and mechanism of action, but they differ slightly in pharmacokinetics and side effects. So they should be chemically similar, should have more or less same kind of mechanism, and have same kind of activity. I'm sorry, did I miss the question? Yes. So this is false because they say which are used for different purpose. No, they are used for the same purpose. Now, what is the primary goal of structure activity relationship analysis? So, A, to determine the molecular weight of a compound. B, to identify how molecular structure influences biological activity. C, to predict solubility of a drug. Or D, to analyze the toxicology of a compound. The correct answer is to identify the molecular structure, how a molecular structure influences the biological activity. So uh, let's try to understand what structure activity relationship analysis actually is. So that's the correct answer, to identify how a molecular structure influences the biological activity of a molecule. So what exactly is SAR? SAR stands for structure activity relationship. It's the relationship between chemical structure of the molecule and its biological activity. Suppose if I have, say, my face, look at my face. So if I have eyes, what's the function of my eyes? What if I remove my eyes? What difference will that make? If I remove my nose, what exactly is my nose doing there? So if I remove it, then I'll realize, oh, I cannot smell anything right now. So that's the significance of having a nose. It helps me smell. Eyes help me see. So that's how we try to study a molecule. We look at the molecule, say, okay, yeah, so there is a double bond, there is this functional group there. What does this functional group actually do? How does this make this affect the activity of a compound? So suppose this compound has, suppose, COH group. So COH group is just protruding out of it. And so what is the significance? If it's an antibacterial compound, what COH does to impart the to to give the compound its like activity? So how is this affecting the antibacterial nature of the compound? So yeah, that's basically what SAR means. So we'll see inside see and try to understand that in a more scientific term. SAR is a relationship between chemical structure of a molecule and its biological activity, how well it works as a drug. It helps us understand which part of drug molecule are important for its action and how changes in the structure affect its potency, effectiveness, and safety. SAR studies help in designing better drugs with fewer side effects. So suppose you understand, there's a big compound and it has a good activity. So by studying every part of it, by studying the significance of what a single functional group is doing for that compound, if that's not increasing its activity, maybe we can just remove it. Maybe because maybe if it's not actually directly contributing to its activity, what if we just change it? What if we remove it? That will make the compound smaller, maybe easier to deal with. If it's not somehow affecting its activity, that would mean that we don't exactly need it. So we can just try to remove that thing or maybe add different thing to increase its activity. So that's how you basically work with the compound. It helps drug discovery by identifying which chemical modification can improve the drug function. Obviously, so suppose there and uh, there's a compound. and you found a chemical modification that, yeah, having SH bond in this compound increases its activity. And then there is another compound, it does not have SH bond, it has different functional group. That functional group might not be, you know might not be involved in, might not be helping it in its activity. Maybe it's just an auxiliary, it does not do anything, it's just hanging in there. Maybe, so what we do is we remove that part, because it's not affecting the activity, it has nothing to do with the activity, it's just hanging there. So we can actually remove it, and we can just, you know just to understand and just to check, maybe try to add SH bond there and see if adding SH bond there is increasing its activity. In most cases, it actually does. Maybe it's lowering down its toxicity, maybe it's somehow helping it interact or fit better in the target site, who knows. So that's how people try to you know fine tune the molecule once they know the structure activity relationship. It helps reduce unwanted side effects. Obviously, once you know that what is absolutely necessary, what makes the compound have the activity that it does, you can just try to add and remove parts and bits of it, and try to add like things to it to make its activity better, to reduce its toxicity. And also it saves time by predicting which modification will work better than actually trying to make the entire molecule and see, okay, so you've added this, and now you're trying to add different thing and wait for the activity and everything, and then you come to know, oh, after doing this much of synthesis, the molecule is actually losing its activity, it's becoming, from a drug it's becoming to a useless compound. So it actually helps us save time and money as well. And how does that actually work? So again, the lock and key model. Imagine the drug as fits perfectly to the lock, that is the target. And if the lock is too big and has extra part, it may not fit. And if it's too small and some parts is missing, it does not fit properly, and hence does not work properly. Structure activity relationship helps us find which part of the key is important, which can be changed without breaking the function. So the steps, that people go through for SAR analysis is, first they try to find out what exactly is the pharmacophore. Pharmacophore is the part that interacts with the biological target and has the activity. So they try to find out, oh, this is the pharmacophore, this is the active part of it. So that's how, you know we say, oh, this is the most important part, we cannot, work without it, this has to stay there. So in case of aspirin, the acetyl group is important part for its pain relieving effect. Now, then what they do is modify the structure, small changes, like say they reduce the double bond to single bond, or maybe they add a double bond to a place where it was actually single bond, to try to make it more rigid, and try to see how that affects the drug action, changing small groups like from OH to methyl, maybe uh, to change the polarity of the compound to see how it affects the activity, and then check the new versions of it. So each modified molecule is tested, and so you had one molecule that was working well, so you modified it, you added another, say, acetyl group into it. Now you want to check, okay, so this is the original one, this is working, say out of 10 it's working 10, 10 on 10, so it's like very good. Now you want to have a drug that works better than 10. So after this modification, does it work just like the previous one? Is it 10, is it 12, better than the one that we had before, or is it lower? So changing and tweaking bits of it, and then trying to compare it with the original one, and then you compare the results. to see which one is the better drug and which kind of modification is working in favor of the molecule, whether it's making it good, even a better drug, or whether it's decreasing the efficacy of the drug. So that's how they do it. For example, let's see the SAR of penicillin. So penicillin, they what we knew about penicillin. It's it has a beta lactam ring, which is very important for its activity. Scientists did a SAR on penicillin, and they modified penicillin to create amoxicillin, which is much more effective and stable. You can see these parts are the same, right? You can see till here it's the same for penicillin as well as amoxicillin. What they did was they changed this. They saw, okay, maybe after changing this part the activity will increase. They went through various different things and finally decided on this. It increased the polarity. Instead of O being here, the O was here. It increased a hydrogen bond. It made it a better hydrogen bond donor, OH, and also acceptor. So you can see here SAR of amide and carboxylic acids are involved in binding. Here you can see that this is amide, and CONH, and this is the carboxylic acid part. These are both involved in binding. Carboxylic acid binds as the carboxylate ion, so it is ionized, COO minus. And the mechanism of action involves beta lactam ring. This beta lactam ring is very important. This entire class of molecule falls under beta lactamase, the enzymes that work on this. So mechanisms of action involving beta lactam ring, activity related to beta lactam ring strained, subject to stability factors. Obviously it has to be strained. And bicyclic system increases beta lactam ring strain, because see, this is a very strained system. We can see that it's a 4-membered ring. Not much variation in the structure is possible because these parts are like absolutely important. That is why they kept this part as it is. Not much variation in structure is possible, and variation is limited to the side chain. That means this is absolutely necessary. You can change this part, and what they did was they changed several parts. So this part is R. For R they added methyl. They tried many different variations, and they found that this variation that made amoxicillin is the best. So that's how they came up with a better and improvised version of already existing antibiotic, which was penicillin. Same was in the case of aspirin. I haven't added the SAR of aspirin, but they came up with different. So they did SAR on aspirin as well, and for beta blockers, and that's how they came up with different medicine like paracetamol, ibuprofen, and for beta blockers they came up with propranolol. So that's how they come up with different and a better drug. So key takeaway is SAR helps us design better drugs by understanding how chemical structure affect the function of the drug, and how small changes in molecules can make drug stronger or weaker or safer. And real world impact, many life-saving drugs like penicillin, aspirin, and beta blockers were improved using SAR studies. Like aspirin, we got ibuprofen, paracetamol with less toxicity, penicillin and amoxicillin and beta blockers. So yeah, that's how SAR helps us design better drugs, and it's very important, like inexcusable part of drug development. So any question from today's slide? Do you have any question for? Yes. Mixed combinatorial synthesis. Yeah, okay. It is actually a quite a messy and, you know, uh, so combinatorial synthesis, what we do is, so uh, these are all based on reasons like surface solid, supported supported reactions. So suppose you have bead A supported, so something like this, like supported, this is a resin and you have starting material attached to the resin. Suppose you have A, B, C, D, and these can be any kind of reaction. Suppose they are all amino acids. Suppose you have five sets of amino acids. What you do is you put in in a vessel, you put all five of them, and you let them mix. After that, what you do is take a part of it and split them in five different reaction vessels. How you do? What you do is, again you put them five, put them again with each other and with everything else that you have, like five, you had five sets, and now you're making again, you're putting everything, you split them. After mixing, you split them, and after splitting, you again have the five thing, and you're trying to mix it again with all this. So you're basically working upon the fragments that you had. You're making working mm, making the blocks from the same blocks that you had. So you mixed five, and you have five different reaction vessels where you have mixture of all five. Now you're adding more to it, more to it. That's how you keep on going. You mix and then you split and then again you mix. So you keep on mixing just to create large library of molecules. And suppose so you don't know in which order they're mixing, because you're just basically, you don't know whether a, that you're having a b c d e, or maybe a b b c c d d, or maybe a b c d and then a. You don't exactly know the sequence in which they are forming or anything. You do this just to create large library of molecule, and in the same vessel, just to see whether this will have some kind of activity or not. Suppose you putting a protein there, if it reacts with that, then we say, oh, it has some kind of activity. Maybe we can slowly try to figure out which compound is of our interest. That's how this, it's very messy, and that's how the combinatorial synthesis works. Is that clear? Okay. Do you have any other question? You can just unmute yourself. Do you have any question from, regarding any of the things that we discussed today? So if that's okay, okay, I'm so sorry. Please, um, yeah, it's okay. So if there's no other question, I'll end this session today. Thank you for joining. Good night. Bye.